Okay, awesome, amazing AP Bio kids. We're ready for parts B and C of the AP Cell Division Lab. So I've printed out, I have in front of me the protocol or the lab directions for parts B and C. In class, when we did this lab, we utilized the microscope. There's the handy dandy compound light microscope. We also used, as you can see here, slides of allium. I'll put it like that so you can see it. Allium, which is an onion root tip. And you can see the 3 roots that they focused on. So anyway, we looked at this under the microscope and we found this. This is allium root tip on high power. This is 400x, okay, on the 40, which is the high power objective lens. But remember the eyepiece is times 10. So this is what they saw. Okay, so for part B, what you have to do, and I'm gonna tell you guys how to revise this since you're virtual. The idea for part B is we want to look at how many cells that we see in one field of view. So we're going to use this as like field of view 1. We're going to use another one as field of view 2. And then I'm going to use a third one of these as field of view 3 for y'all. And what you're going to do, I'll zoom in on it and kind of like, you know, let you take a snapshot of it. But you want to count how many cells are in interphase and how many cells are in M phase, remembering that M phase would be P, P, M, A, T, and cytokinesis. So it's really interphase versus anything in M phase. And you want to count how many total cells that you see in the field of view. So we're going to do this 3 different times. The reason that we're doing this in 3 different times is because we want to do a wide range of data. We want to get a lot of cells so that we can get a total number for data collection purposes. And then I want you to find the percent of that. We want to find what percent is in interphase, kind of total out of 3 fields of view, what percent is in M phase, because this gives us a better idea on one slide how many total cells may be in interphase versus M phase, okay, dividing. And so once we do that and you calculate the percent, then we can use that to do some standard deviation and do some data analysis. Okay, so we're ready for our first field of view. So you're going to record your data here, and this is going to be your field of view number 1. Okay, so I'm just going to kind of focus on that. You may want to pause and do your counting, but just to give you an idea, because I know you guys aren't here for me to help you with this, if I just start right here in the bottom left, do you guys see how, by the way, the dark circles are the nucleus. These are plant cells, so you can see the cell wall right here. See how that is nice, beautiful cell wall. And so the nucleus in this picture, it's still kind of intact, but do you see how there's lots of white space in the nucleus? This particular picture right here, this is prophase. So what you can see is the disintegration of the nuclear envelope in this area right here, and the DNA is thickening and coiling and condensing. So this is a definite prophase. So that would count for 1 over here when you're doing the M phase. That counts for 1 cell that's in M phase. If you look at above that, can you guys see that this is a nice little cell plate right here? This is 1 plant cell, what I'm outlining with my pencil, and it is in telophase and cytokinesis. So that means this one's in prophase, this one's in telophase cytokinesis, so that's 2 so far that are in M. All right, look at the one above it. You see this one? I also consider this prophase because I can see the coiling and condensing right there where you see my pencil is. Okay, so that's a 3rd one, that's for M. If I continue to go up, okay, look at this one. This one is kind of like really light, almost hard to tell. When they're hard to tell like that, that's interphase because the cell is just, you know, it's just there. It's in chromatin form, not doing much. The one above it, same thing right here. So that's 2 that are in interphase. Now, when you get to something like, let's say, let me see, this one doesn't have any dead cells. A lot of times you'll see dead cells. Well, this one right here. See this one? You don't really see anything. That's a dead cell. Don't count that one. Okay, but if you see like this one right here that I'm pointing to, that's prophase. So then you may be saying, well, what does interphase look like, Miss Lynham? Look at this one right here. Classic interphase. You can see the nucleolus in the center. And so that's interphase. Over here, that's interphase. Okay, let's see if I can see another one. Right here, that's interphase, even though it's a half a cell. So use this for your field of view number 1. You're not going to be counted off if you're a couple cells off, guys. Okay, that's not how I'm going to be grading this. So just do your best and use this picture for your field of view number 1. Okay, your field of view number 2, I want you to use this image. Okay, now this one has a lot of dead cells. Look at this. Don't count any of these. These are just dead cells. And if it's really hard to tell, like I would say this is interphase, even though it's lightened right there, interphase for these, but these are definitely empty, so those are dead. So use this one for your field of view number 2, and then use this one for your field of view number 3. Look at that beautiful, y'all, that is such a good pro-metaphase right there. Sorry, I just had to comment, look at that, that's pro-metaphase. The nuclear envelope is completely gone. All that DNA is thickly coiled and condensed, and they found each other as sister chromatids, and next thing they're gonna move to the middle of the cell. That's a beautiful picture. Okay, so that's field of view number 3. All right, once you do all your counts for field of view 1, 2, and 3, record in this area, total up your number of cells, and then find the percent of each. Okay, does that make sense? All right, after that, because that's pretty much it, the only other thing you're gonna do, this has nothing to do with the comparison of how much time is in interphase versus M phase for our data analysis part of our lab. This is really, I want you to use this formula down here at number 7 to estimate, according to the number of cells that you see, what's the approximate number of time that they're gonna spend in each of those phases. So there's a little formula for you to use right here at number 7 to enter that. Okay, since I'm on the topic of that part of the lab, I want to switch now for a second and show you guys the write-up guidelines. This is the write-up guidelines for the AP Cell Division Lab. So it's a typical format that I normally do, introduction, materials and methods. Part A, you've already completed your drawings, so there's really nothing for you to do right here in your lab notebook. You'll start B and C, which is your data tables that I just showed you, but this is the section I really wanted you to look at right here. The data analysis. Okay, for part A, there's really not much to do other than vocabulary. I want you to use your notes and then kind of break down like short, concise, descriptive bullets, what's going on in interphase versus M phase. This is the part, I'm gonna put a star by this. Part B, data analysis, is what we're using this data for, okay? So if I look at this chart where you recorded the, ooh, my pencil just broke, beautiful. If I look at this chart, where you recorded the percent, the total number of cells that are in M phase versus interphase, we're gonna use that to do some class data and actually graph it and see if there is a significant difference. So my question to y'all, look at it right here, it's in bold. It says, is there a significant difference in the average number of cells in interphase versus M phase in the cell cycle? So what I'm gonna post for y'all is class data. It's gonna look like this. And so you will copy down like the data from table 1, table 2, table 3, table 4. And then for the virtual, I'm actually gonna put Linum on this. It's Linum's data. So it may not match y'all's exactly, but I wanted y'all to have some data to be able to use to represent the virtual kids. So that's gonna go in this section right here. And you'll use this to find the average and be able to use the average to calculate standard deviation and standard error of the mean. So if you're not sure how to do this, remember, go back to some of the other labs that we did standard deviation and standard error of the mean. And then I have the formula on the back right here on the next page. So that's what you're doing with the math part of counting these cells. Okay, now for the most fun part of this lab. I'm so sad that you guys can't be here to see this because this is actually fun to use the meiosis beads. But we're going to start part C. It's right here, a comparison of mitosis to meiosis. And what I provided for the kids is this chromosome bead set. Okay, it came in a bag like this. I counted them all out. And so there's enough materials right here on the table to represent a cell, and I drew it on this side, 2n equals 6, okay? So if 2n equals 6, remember that's the diploid number, then that means 3 chromosomes originally came from mom and 3 chromosomes originally came from dad. So I chose pink and blue, but I wanted to show you guys that for every 1 chromosome over here from mom, there is a match from dad. We call these maternal and paternal chromosomes. So for example, look at this one right here. There's 3 beads at the top and then there's 5 beads at the bottom. These represent genes on a chromosome. So if this is chromosome number 1 from mom in this cell 2n equals 6, there should be one that matches that from dad. And you can see right here, this one matches. Okay, so these are matching chromosomes. But this counts as 2 chromosomes. So that's gonna represent what we say chromosome pair number 1. It's like having a pair of shoes, but you have a left shoe and a right shoe. And so one is from mom and one is from dad. Well, I can do the same thing on the next one. Look at this one. This one has 3 genes on the top and only 4 on the bottom. That one's from dad. This one's from mom. Okay, so that represents chromosomes number 3 and 4. The final one, okay, look how many genes are on this one. One from mom, one from dad. So a word that we use in class, what I say is, this chromosome from mom is homologous to the one from dad. The term homologous means contains genes in the same place that code for the same traits in the same position. Same loci, same genes, same traits. So those two are homologous, but they're not identical. One's from mom, one's from dad. Okay, well, if we're going to perform, let's say we go through cell cycle. So this is going to be in a somatic cell, aka skin cell, in the body, because that's the one that skin cells are the ones that go through the cell cycle most frequently. Look what I have on the table. I'm in G1 right now. Okay, G1, 2n equals 6, and I have 6 chromosomes. Well, if the cell goes to S phase, look what I'm going to do right over here. All of those chromosomes are going to double. Oopsie, one of my chromosomes just broke. There we go. They're going to double. Once all the DNA doubles, and remember, we're in interphase, so it's in chromatin form, right? It's all loosey-goosey, jumbled up DNA, looks like stringy spaghetti wrapped around its little proteins, but it's all loosey-goosey. So after the doubling, we move to G2. G2, remember, there's nothing with the DNA that's going to happen. We just replace old and worn out organelles if we need to. Okay, here's the cool thing. We're going to click into prophase. As we click into prophase, here's my nuclear envelope. We're going to put some cracks in it, some holes like this. As we put the holes in the nuclear envelope, the most important thing in prophase is the DNA starts to thicken, coil, and condense and find sister chromatids. So here's the original maternal, okay, chromosome. It's going to find the identical copy. That's known as sister chromatids right here. There's 2 chromatids to 1 pair of sister chromatids. But there's also the paternal one right here that was homologous, and it's also going to match up with its sister chromatid pair. So those are matching up like that. I'm going to do the same thing for every one of these that finds its little, is that the right one? Let me find all the right ones. All right, so all my sister chromatid pairs have found each other. And you can see 3 from dad, 3 from mom. Sister chromatids are all attached at their centromeres. That's what this represents, centromeres. And now we're going to move from prophase to prometaphase. Well, if we go into prometaphase, we're going to get rid of this nuclear envelope even more. It's almost completely disintegrated. Prometa. Now, I don't have spindle fibers here. Actually, I do have a couple that we can show. These little spindle fibers, aka microtubules that are kinetochore microtubules, they're going to grab on like this and start to move each sister chromatid pair and kind of get them closer to the middle. So each of these kinetochore microtubules makes a connection, starts to move them to the middle, kind of like that. Okay, you with me? All right, once we go to metaphase, these are going to line up in the middle of the cell. Call that the equator, right, or the metaphase plates. And there's really no particular order. They just line up, and it'll look something like that. So they're lined up in the middle of the cell, and there is no more nuclear envelope present because metaphase literally just has them lined up to do a head count. Okay, from that point forward, we're clicking into anaphase. So all the kinetochore microtubules grab on and go like this, and pull the sister chromatids apart. So it'll look like this. This one pulls like this. This one pulls like this. Okay, this one, same thing. And so they all pull apart in anaphase, and they're moving to opposite sides of the cell. So when we're done, let me show you the result. When we're done, we now have on this side 1, 2, 3, 4, 5, 6. These are daughter chromosomes. On this side, 1, 2, 3, 4, 5, 6 daughter chromosomes. They move to opposite sides of the cell. My new cell over here is still 2n equals 6, and this new cell is 2n equals 6. So I have 2 identical cells that have the exact same number of chromosomes and same amount of DNA. And that represents cell cycle. How cool is that? Okay, so now for the tough one. We're ready for meiosis now. Instead of producing somatic cells that are diploid at the end, we want to produce haploid sex cells. The intent is to produce sex cells for the purpose of fertilization. So I'm still starting with my original cell. This is a germ cell. Okay, don't forget, let me write this down. the original cell is a germ cell in the ovaries or testes, and it is still diploid. That's the important thing. We're starting off with a diploid germ cell. When we are done, we want to end up with 4 haploid cells. And look at this. We want it to be half of 2n equals 6, so we want n equals 3. All right, but here's the deal. Look what this looks like in G1. Do y'all see this? This is G1, which means for me to have n equals 3, I need 3 singles just like this. This is 2n equals 6, but they're single pieces of DNA. I need n equals 3 single pieces of DNA. All right, so let's roll. Here we go. We are in G1. We click into S phase. We do exactly what we did if this was a somatic cell. We replicate. S phase of interphase, replication of DNA. There it is. It's all chromatin. Everything's great. We click to G2. G2, we double all organelles, exactly like we do if we're performing the cell cycle. All right, for the most important, we click into prophase 1 of meiosis. Prophase 1 of meiosis, in my opinion, is the most important step of meiosis because that's when we're going to, number 1, get homologous pairs, or the tetrad that forms, and number 2, we're going to establish genetic variation. We're going to cross over some chromosomes. So I'm going to show you what happens. Let's say we're talking about this one. Look at this long chromosome. Okay, that's the original, okay, per se, and it's going to find its sister, look at that, sister chromatid. But because we're not making more skin cells, this is paternal, you can tell it's all blue, it's gonna find the maternal sister chromatid pair that's just like it. All right, look at this. I've got, this is the original, there's its replicated copy, this is a sister chromatid pair. Here's the original, same chromosome, but from mom, and there's its sister chromatid pair. Well, because these represent the same chromosome, let's say number 1, these are going to synapse together. And when they synapse together, it's called synapsis, they're going to crisscross applesauce like that, crisscross applesauce, and we say the pink ones right here that are maternal, those are homologous pairs compared to the blue ones. And so homologous pairs come together, and where they cross is known as the chiasmata. That's chiasmata. And wherever they cross, they're going to actually break and switch genetic information. Okay, so the more chiasmata that you see, the more crossing over that's going to occur. Okay, so just for the sake of time, I only crossed over a little bit on these because I didn't want to take up too much time. But do you notice how this is important, that when I look at this tetrad, okay, I'm calling this a tetrad, or I call this homologous pairs are synapse together. Listen to the vocabulary. The thing that I notice is the paternal, the blue one, is still stuck with its paternal. They're still stuck together at their centromeres. My maternal are still stuck together at their centromeres. Same thing over here. Paternal stuck together, maternal stuck together. Maternal, paternal, and it doesn't matter, like these could go that way, okay, where maternal's on the right, paternal's on the left, or they could be this way, maternal's on the left, paternal's on the right. And that just also leads to more variety, more variation, because it kind of doesn't matter which way they lay. They go different directions. But like I said, the important thing is paternal stay stuck at their centromeres. Chiasmata is where they cross with their homologous pairs, their homologs from the other parent. So you are looking now, that's prophase 1 of meiosis. The rest of this is pretty easy because if we move from prophase 1 and go to prometaphase, they start to go towards the middle. If we go to metaphase, it's going to look like this. But like I said, more variety is they could lay with maternal on the right, but like this one, maternal's on the left. So that's metaphase 1. When they go to anaphase 1, they're going to pull apart, check this out, but the maternal originals still stay stuck as sister chromatids. Paternal originals stay stuck as sister chromatids. Same with this one, same with this one, same with this one, same with that one. So we're going to get 2 new cells. This is going to be in one cell right there. This is going to be in another cell. And technically right now, this is where kids get this confused. At the end of meiosis 1, we are really haploid and we are really n equals 3. Look, 1, 2, 3. That's half the number of chromosomes. That's half of 6. But the problem is at the end of meiosis I, we can't stop here because they're still stuck together as replicated chromosomes. So this was the original that's still replicated. Same with these, same with these. So we still have to go through a second meiosis. We need a meiosis II so we can take these sister chromatids that are still paired up and pull them apart all the way so we can get cells that are n equals 3 but have half the amount of genetic information compared to what was in G1 of the original cell. Okay? Okay, now for meiosis II, this one's really easy. We have, this is in one cell, this is in the other cell after meiosis I. So I'm just going to show you on this one, but it's going to happen in both cells because remember after meiosis I we get 2 cells as a result. This is going to move directly into prophase II. Prophase II, sister chromatids still coil condense, wrap around their histone proteins and look like this. Metaphase II, check this out. Metaphase II, these are going to line up like this. They'll do the same thing in the other cell. Metaphase II of meiosis, they line up like that. Notice how they're all jumbled up. They move to anaphase II, pull apart, pull apart, pull apart, like this. And now when we go to telophase cytokinesis II, these 3 chromosomes right here end up in one cell. These are 3 different chromosomes, but look at them, y'all. It's some pink, some blue, some blue, some blue, some pink, pink and blue, and pink and blue. They're all jumbled up due to the crossing over, but I want you to pay attention. It's one single chromosome. Now we are at n equals 3, and we have single chromosomes. And if you're like, what? Y'all, this is gonna be a sperm or an egg cell, right? Let's say this is a sperm. Well, this sperm is gonna join with the egg, which is also n equals 3. So my new zygote after fertilization is going to get these 3 from, let's say, a sperm, and they're going to get the other 3 that match those from the egg, and then the zygote, the new offspring, is going to resemble the sperm and the egg, which is the mom and the dad. Okay, so pretend like these are the 3 that Bob, we're going to call the sperm from Bob, right? Bob's sperm. And let's pretend like Bob is going to have sex with Sue. And these are the 3 chromosomes that Sue is bringing from her meiotic process. The new baby, whenever these 2 meet, watch what happens. This one is going to meet up with that one. That's 1 pair of chromosomes for the new baby. Let's say that represents chromosome pair number 1. The new baby gets Bob and Sue's sperm and egg DNA, sperm and egg DNA. The new offspring after fertilization is going to get 1, 2, 3, 4, 5, 6, 2n equals 6. But now it's one of these from the parents, and now this starts over, and this is the new baby's DNA. Isn't that amazing? That, my friends, is meiosis.